transcription. Now, how do we actually make a protein? The process is called translation. Okay, so go ahead and write this down. Genetic code. All right, so this is how the body actually knows what protein to make based on the DNA strand. It's kind of like binary code, except for you have 4 instead of just 2, and you're making proteins instead of making computers do work. mRNA contains a code using A's, U's, C's, and G's. The complete instructions for building protein, and code must be converted to protein language. Actually, you don't need to copy that down. Let's take a look at this, okay? So this chart right here, this isn't something that was just created out of nothing. This is done with, years and years of research. Basically, trying to figure out what amino acid does the body equate to the mRNA strand. So write this down. A codon is a group of 3 nitrogenous bases, and they code for 1 amino acid. We can see on this chart here, you have to have 1, 2, 3 letters to be able to make 1 amino acid. Okay, here's an example. UUU codes for, I don't know how to say that, phenylalanine placed in a protein. So let's go take a look at the chart. So here's U here, and then on my third letter is U. So right there, PHE, we know that codes for the amino acid phenylalanine. And what we basically have here, that's 1 amino acid, which is not a protein. A protein could be, thousands of amino acids. So that would just be 3 little nucleotides off the DNA or mRNA. They would code to 1 specific amino acid. You have stop codons, UAA. Let's follow that. That just means that the protein is ending into stop. That means the protein is basically done, and then you need to stop so it can fold into place. Let's see here, UAA, UAA, yep, stop. Okay, we'll get some practice with that in class. There are 64 possible combinations, but if you look, there's only 20 different amino acids. Okay, so some of these codes will code for, or some of these amino acids will have multiple codes. You can see there that, I think it's leucine, has like 6 different possible combinations it could code for. And more than 1 codon can code for 1 amino acid. I just said that. Here's just an example. Like I said, we'll go over this and we'll do some practice of this in class, where what you'll have to do is you'll have to take a DNA strand and then make a protein out of it. But this part's basically easy. Once you've already created your mRNA, you take your mRNA and you take a look at this genetic code chart. It will tell you what proteins the ribosomes will put together. So there's C, so we know it's right here. Obviously A, right there, and then U. So we can make the assumption that histamine or histine is what you're going to make. Let's take a look at that. Oh, histidine, I was wrong. I need to work on my protein. Okay, you don't need to write anything on this page, guys. Let's move on. All right, so here's an example here. This would be something that you might have to do on a test or something like that. So you're given the DNA code, 1/2 the DNA strand, right? And then you have to convert it to mRNA. Well, it's easy. The T's go to A's, the A's go to U's, the C's go to G's. The only thing weird is that when you're making an mRNA strand, there's no T's on here. So that's where the U comes in. So A will go to U as its complementary nucleotide instead of A going to T, like if this was another DNA strand. So you have to be careful. If you're making mRNA, there should be no T's on here. So let's make the protein. We know it takes 3 nucleotides to make 1 amino acid. So we got the start codon, we got the cysteine, tyrosine, histidine, and should be a stop codon. So there we go. We made a very short protein, just 3 amino acids long. Obviously, in real life, most of them are going to be way longer than that. All right, now let's talk about translation. So transcription is the first process where we make an mRNA. Translation, let's simplify that down. Make sure you write this down, guys. Second step, mRNA strand is taken to the ribosome, and the ribosome creates a protein. So we're translating the mRNA strand into a protein. So that's why it's called translation. Transcription, we're writing an mRNA strand, basically. Translation, we're decoding it. We're translating it into a protein. So this is the second step, where the mRNA strand is taken to the ribosome, and the ribosome creates a protein. So this is a simplified way to say it. Make sure you write that down. Let's move on to the not so simplified way. So this occurs in the ribosomes. The ribosome attaches to the mRNA like a clothespin. The sequence of bases is converted into a sequence of amino acids, uses the universal genetic code. Every 3 nucleotides on mRNA equals 1 amino acid. tRNA brings needed amino acids to the ribosomes to build the protein. So this is something we haven't talked about, but tRNA, well, I guess we mentioned it, but tRNA brings in the amino acids and basically allow them to form a chain to build a protein. And let's take a look. Go ahead and pause the video and copy all this down. This is kind of the detailed information about translation. All right. Take a look at how this works. Here we have a strand of mRNA. You see it says mRNA. So always know if you're looking at mRNA or DNA, because that's going to be important. Obviously, if it has the U on there, then we know it's mRNA. So this is happening in the ribosome. Here we have a TNA, really, really small molecule compared to the ribosomes. And it's got an amino acid on one end, and it's got what we call the anticodon on the other end. So on the mRNA, every 3 things is called a codon. And then on tRNA, every 3 things is called an anticodon. And we can see here that this has CGC. So what would that would pair up to GCG. So where would that be on here? I don't see GCG on here. So it actually wouldn't connect to anything on here. That's okay though. So here, if there was a GCG on the mRNA, this would snap into that and bring the alanine amino acid into place with the rest of them in a line. So write this down. tRNA has 2 sides, amino acid side, the bottom side, nitrogenous base side, that's where the T shape comes in. It doesn't actually look perfectly like a T, but it looks close. Then you have the anticodon, 3 bases of tRNA. It's the opposite end of amino acid and pairs up with the mRNA bases. So write that down. Pause the video if you need to. Okay, now I want y'all to stay with me on this one. We're going to talk about this picture right here. This is kind of like your photosynthesis cell respiration diagram. This shows a lot of stuff in one picture, so it can get kind of confusing. But we basically have transcription over here, translation over here. How do I know that? Well, just by looking at it, I know that transcription happens in the nucleus, translation happens in the cytoplasm and ribosomes, so I can just make that assumption by looking at it. But let's see what happens here. Once again, this whole process shown in this picture is protein synthesis. Okay, we're just trying to make a protein, but we're going to learn how we do that. All right, so first thing that has to happen, transcription. We have to make our mRNA strand from half of a DNA strand. So there's a gene right here coding for something. Here's the mRNA strand that was created off of that during the process of transcription. Now, that mRNA strand has to be taken to a ribosome. Here it says ribosome. And the ribosome is basically a giant protein that will allow the mRNA to be translated from mRNA into a protein. Remember, the whole purpose of this is to create this protein chain right here. Now, you might think, well, how do we create a solid structure out of a chain? Well, this chain will eventually fold into different shapes depending on the, basically depending on what amino acids was in it. So this protein will not end up like a chain. It'll be a little ball of proteins. All right, so what happens with translation? Well, it's very simple. The tRNA with the right anticodon on it brings in and attaches to the mRNA, and one by one, the amino acids are built into a chain. Remember, these chains can be thousands of amino acids long or just a couple depending on the situation. And we see here that you have free-floating amino acids in the cell. In the tRNA, simply a specific tRNA bonds to a specific amino acid. So remember, there was like 64 different combinations. So you technically have 64 different tRNAs, but there's only 20 different amino acids. So you only have 20 different amino acids floating around. And those one by one will get built up, and you'd have an amino acid chain. Now, this is assuming your body's healthy, right? Obviously, if a cell doesn't have access to amino acids because, let's say, you're starving or just haven't had enough protein, then the cell won't be able to make the protein chain it needs, and therefore the cell will get sick, could potentially die, or just cause some symptoms for the person or cell that organism that the cell belongs to. All right, guys, that's all we're going to do. Make sure you get these notes done. We'll do some practice of this when we come back to class. Thanks, guys.